Let me start by saying that each neuron in the brain has a very distinct electrical personality. Some neurons don't do much unless they're being stimulated. Some can generate various different types of electrical patterns, and it's really a massive number of different ways that a neuron can fire, even when it's not being stimulated. And so I'm going to start off by giving the very basics of what it takes to generate these impulses in the nervous system. The major factors that determine the firing of a neuron are the sodium channels that give you the upstroke of the action potential, so that when a neuron is stimulated, sodium comes in through the sodium channels, and there's 3 genes that encode most of the sodium channels in the central nervous systems. Those are NAD1.1, NAD1.2, and NAD1.6. And as many of you know, mutations in these genes can also give rise to Dravet syndrome or other epilepsy. So there are not many genes. There's 3 main ones in the nervous system. There's only 9 altogether. And they're responsible for the upstroke of the action potential. Recovery from the action potential, and in fact, anything that then restores the membrane potential or keeps the membrane potential negative, is largely the function of potassium channels. And there are over 70 different genes that do this job of repolarizing the impulse of the nerve impulse. So we're just going to focus on one of these, and that is SLAC, also known as KCNT1 and KNA1.1. There are 2 genes that are sensitive to intracellular sodium, and that's what makes these 2 genes a little bit different from nearly all of the others. The others sense voltage, they sense calcium, but these 2 sense the amount of sodium on the inside of the cell. And they're both very big proteins. The first one that we'll be talking about is KCNT1, SLAC. SLAC stands for sequence-like a calcium-activated K channel. That name was given to by Bill Joyner, who first studied it in our lab. This has an intermediate conductance, which I'll explain later. But so when the second one was isolated by Aaron Bhattacharjee in our lab, he named it SLIC, or sequence like an intermediate conductance channel, and that's KCNT2. So we can record the electrical activity of these channels simply by expressing them in a really boring cell that's not a neuron, so that when we do electronic wizardry to measure the currents that flow across the membrane of the cell, we can measure only the current that flows through these particular channels. So this is an example of current flowing through a KCNT1 channel. The SLIC channel on the whole activates more rapidly when you jump the membrane potential to a positive value. And the bottom slides show the opening and closing of single channel proteins using a technique called patch clamping. Each of these little jumps represents measurements that we can make of a single channel just opening and closing. And the way we do that is by a technique called patch clamping, where you put a fire-polished electrode on the surface of a cell, and if you're lucky, when you make that seal on the surface of the cell, you will end up with, or sometimes 2 or 3 or 4 different or single ion channel proteins under the membrane. And when you do this, and you happen to have SLAC under there, then you can manipulate sodium on the inside. And what you see on the right-hand side of this slide is the opening of these individual SLAC channels. I'm going to call them SLAC because that's what most of my slides say, but you can put KCNT1 in your brain and it's the same thing. So these yellow traces, when it's just a straight line, means there's no channel openings. These individual deflections to upwards here represent the opening of single proteins, the channel becoming active. And you can see as you increase the amount of sodium, the channel becomes more active. So when does this happen in real life in a neuron? Well, the first obvious time it happens is that when the neuron generates a nervous an action potential, an impulse, and sodium enters through the sodium channels. And that's an obvious way that these channels become active. The second way that's been shown that they can become activated is during excitatory transmission. When an excitatory transmitter is released from a presynaptic engine, it opens glutamate receptors that let sodium into the cell, and that can also activate SLAC channels. Finally, there's a third way, which I'm not really going to talk about, but it's actually a very interesting interaction. There's a class of channels called HCN channels that are hyperpolarization activated channels, sodium channels. I'm told that Justin is waiting in the lobby. I'm going to let him in. Oops. These actually play an interesting role in working memory in the frontal cortex. And this is a specialized relationship between these two channels, which I can talk about if anyone's interested, but I'm going to leave that for now. So the SLAC gene is a really big, complicated gene. Actually, the protein itself is pretty big and complicated, but there are, in everyone's DNA, there are 31 different regions that have to be joined together. They're all separated along the DNA, but they get linked together to form one messenger RNA, and those are represented by these thick bars along the DNA here. In mice, there are two starts of transcription. That means there's two different places in the genome where you can initiate the synthesis of messenger RNA for SLAC channels. And then these various exons are where parts of the protein get put together. So we've done a number of manipulations, or we and others have done different manipulations of this gene to be able to study what SLAC channels do. And the first is simply to delete exon 11, and that is where the main part of the channel, the part that lets through the ions, resides. So if you delete this exon, you will have no functional channels. However, as we have discovered, that doesn't stop the animals still making at least some proteins that look like SLAC, even though they're not functional channels. So another approach we've done, and we now have animals in which we have deleted the starts of transcription. That means there is no functional protein whatsoever. Finally, mutations that cause disease, that cause the various epilepsies, they tend to be on these other exons along here. And exon 15 is where there's R455H, which is the R474 mutation that we've used a lot in our animal models. Finally, we've actually made a mouse in which we've taken the whole human genome and put it into exon 3, the whole human, sorry, not genome, the whole human sequence of KCNT1. So under those conditions, the mouse now only makes the human channel. It's not making any of the mouse proteins. So that's something we're just beginning to study. All right, so let's start with what happens when you just delete the pore. So you don't have SLAC channels. The animals are not as inquisitive as their litter mates. So if you put a wild-type mouse into a cage in a strange environment, it starts looking around, looking at its environment, and then over a period of an hour, it gets used to what it's seeing and it settles down. And it's not resting much, and it sort of rests towards the end of an hour. In the SLAC knockout, which is missing the pore, it's not as excited to find itself in a new environment, and it basically rests the same amount of time at the beginning and at the end. The other problem that they have is that they don't do well in procedural learning. And this is tested by putting a mouse on a rotating rod. And when you first do this, you put the mouse on and it falls off. But if you keep putting it back on, it learns, like learning to ski, it learns to stay on the rotating rod. And you can see that with a normal mouse, tested every 10 minutes, and it gets better and better throughout the day. And then you put it back on that rotating rod the next day, and it's doing perfectly well. It remembers what it did the previous day. In contrast, the SLAC knockout mice, they don't learn to do this, and obviously the next day they know better because they didn't learn it in the first place. All right, so they have other learning problems as well. That they once they do learn something, they're capable of learning, but they can't undo that learning. So that's okay. Perhaps a more severe problem with the knockout animals is that they don't have synaptic plasticity. So the two major forms of synaptic plasticity that people have used as proxies for what it takes to learn something are long-term depression and long-term potentiation, usually in the hippocampus, but you can study this in other regions of the brain. And what happens is that you stimulate a synaptic pathway, and if you stimulate it once a second, but then stimulate it once every 20 seconds, and then once every second, that's a low rate of stimulation, and the synapses gets weaker and weaker. And then they stay weaker for hours after that training paradigm. But in the SLAC knockout, they just go back to where they were. The same thing with this very well-studied phenomenon of long-term potentiation, LTP, where if you stimulate the synaptic pathway in the hippocampus at a rapid rate, 100 hertz just per second, the synapses become much stronger than they were, and that lasts for hours. But in the case of the SLAC knockout, nothing happens. They don't get any stronger. Now, this is not our work. This is Peter Ruud and Rakowski in Germany. But we have also looked at this. And the way we've looked at it is that when you stimulate one of those pathways, the reason the synaptic strength becomes greater is because you insert new transmitter receptors into the plasma membrane. And there's a way of doing this in cultured neurons where you can simply visualize the insertion of the receptors into the plasma membrane. So this is staining for the GluR1 type of synaptic receptors. And then we mimic what happens during LTP by giving a compound that stimulates the main receptor that's activated during this potentiation process. And this is staining on the outside of your cell. So you can see under normal conditions, when you stimulate the cell, it puts in all these receptors onto its plasma membrane. That's quantified on the bottom left for the normal neurons from the normal animal. And you can see you get this marked increase in the intensity of these receptors on the plasma membrane. If we simply give a drug that blocks SLAC channels, that no longer happens. It also doesn't happen if we give a drug that is well known to block long-term potentiation. But not only does the drug block it, but the animals that don't have the functioning SLAC channel, they also don't show this phenomenon at all. So we're actually spending a lot of time looking at the mechanism by which this is happening. Okay, I'm going to stop there for 1 second in case anyone has any questions about what I've said, because I'm going to leave the SLAC knockouts for a while now and move on to the gain of functions.
Len, I have a quick question. This is Al George at Northwestern. Very nice work. The question I have is about whether the heterozygous knockout SLAC mice have the same dislearning phenotypes. Is that known?
We're actually looking at that. Preliminarily, yes. But we have one experiment in which we've done that with the heterozygous. I'll show you some other commonalities at the very end between the het and the knockout, but that we don't fully understand. But yes.
Thank you.
Okay, so now let's talk about the SLAC protein itself. So it's the largest potassium channel there is. One of the isoforms is bigger than the other biggest channel that's known as the BK channels. And this is a published structure from Rod McKinnon's lab of the SLAC channel. And it's a little bit misleading because you can't get the structure for the whole channel. Only 1/6 of the protein is actually the membrane spanning regions shown in the plasma membrane here. The rest of it is a very large cytoplasmic domain, and this is the structure shown of that. But there are still an additional 200 amino acids on the end of the C-terminus that are not shown here because they don't have a structure. And there are additional amino acids at the N-terminus that also are down here that are not shown. And the interesting thing about this very large C-terminus is that it binds some very interesting proteins. So one of the proteins that it binds is called factor 1 for phosphatase and actin regulatory protein 1. This brings a protein phosphatase to the channel. It regulates its phosphorylation, and it also links the protein to the protein G-actin. More curious, however, was the finding that it binds two proteins whose function had never been thought to be in anything to do with the regulation of ion channels. The first of these is FMRP, the Fragile X mental retardation protein, which is an mRNA binding protein, which main function is to regulate the translation of a subset of messenger RNAs in the brain. The second protein it interacts with is CYFIP1, the cytoplasmic FMRP interacting protein 1. This is also a regulator of protein translation, and even though these both have independent functions, they come together a complex, which is why this has the name of cytoplasmic FMRP interacting protein. This was initially a surprise, but now there have been multiple publications showing that all these channels on the right also interact directly with FMRP. We can co-immunoprecipitate the channel with FMRP. We can actually, when we pull down the channel, we can pull down messenger RNAs that are bound to the protein. And this is a technique called force resonance energy transfer, showing that they interact very closely. And for years, this has been described as a non-canonical function of FMRP. In other words, that in addition to regulating FMRP is regulating the channel. Well, we think it's not just a one-way conversation, but I'll show you now that FMRP does regulate the channel. This is, again, single channel recordings where we've actually pulled off little bits of membrane that have 3 or 4 SLAC channels in them. And the reflections from the baseline here reflect, again, the opening of single KCNT1 SLAC channel proteins. So most of the time you can see one opening, but sometimes 2 or 3 open at exactly the same time. And what's obvious is that actually if you add FMRP to the inside of the patch, now the channel becomes much more active. You get many more simultaneous openings of channels. When you measure the total current, it goes up. In animals that don't have FMRP, the amplitude of that current is reduced, as shown on the right. A little tidbit for people who are really into channel biophysics is that the normal KCNT1 channel, when it doesn't have SLAC there, has a hard time deciding whether to open or not. And it goes into these what are called subconductant states. But in the absence of FMRP, those subconductant states completely disappear. So you get very clean openings to 1 opening, 2 open channels, or 3 open channels. And these little histograms showing you, just add up all these points, these are the subconductant states. So, but also if you inject FMRP into a neuron that has SLAC, you get increased repolarization, exactly as you would expect for an increase in potassium current. And if you inactivate the FMRP, nothing happens. Okay. So mutations, as you all know, in KCNT1 are devastating in many ways. And this shows a number of these mutations. These are all mutations that we've looked at in our lab. I think over the past several years, there's now been over 70 papers describing about 60 different mutations in KCNT1. And the major condition is this epilepsy of infancy of migrating focal seizures that starts within a few months of birth. It used to be called malignant migrating partial seizures in infancy. There's another condition called sleep-related hypermotor epilepsy, SHE, also used to be called AD and LFE. And the interesting thing about this is that there are a number of mutations that can cause this pattern of seizures, which occur later in development, between ages of 6 and 8 generally. And they don't need to have intellectual disability. But if you have a mutation in SLAC that causes this, there's a severe intellectual disability even before there are any seizures going on, which makes us believe that the mutations that cause the problem here are causing the problem not simply because they're disrupting the pattern of electrical activity giving seizures. There's something else going on. Ohtahara syndrome has been reported, infantile spasms, and a number of other conditions have been associated with SLAC mutations, but these are the two main conditions. And they are just about all gain of function mutations. And there's many different ways in which you can get a gain of function into the SLAC channel. These are the first two we looked at, and they give you a 3 or 4-fold increase in the amount of current. So this is where we take the messenger RNA, express it in an oocyte of a toad, and measure the currents, and you can just measure the currents going through the channel. And with these T mutants, you can see they're much bigger than the currents that you get with the wild type. And they uniformly at all the voltages tested, they are greater. These two particular ones seem to increase their amplitude by mimicking the action of a protein kinase. So we know that there's a volume switch on the activity of KCNT1 channels. And that's this serine 407, which is situated in the cytoplasmic domain, just south of the membrane spanning regions. And when this is phosphorylated, that increases the amplitude of the current. There's an enzyme called protein kinase C that you can stimulate with this drug called TPA. And when you normally apply that to a wild type channel, you get this marked increase in currents. It's stimulated the activity of the channel. With these mutations, and just showing you one of those here, you already fully max out the activity of the channel. And when you try to further stimulate the activity of the kinase, you get no further increase. So both of these mutations, one of which is actually right next to the phosphorylation site, that increases the activity of the channel. But that is not the only way that the currents can be increased. In some of the mutations, in particular the one in the R4558, the 474H in humans, that increases the current much more than you can increase just by the protein kinase. You can see that on the top, these are a number of different mutations compared to the wild type, and there are very large numbers, very large increases with all of these. Although it's quite variable, here are the ones that we've looked at before, the first ones we looked at, but some of these have much greater increases. So as I said, that some of them can give rise to increased sensitivity to sodium, some of them have increased sensitivity to voltage, they open at more negative voltages, but it doesn't look as though any of those can fully explain why there's such a big increase in current. And so what I'm going to talk about now is the way that the gating, the opening and closing of these channels can be influenced by neighbors. So the default position for an ion channel, when you think of an ion channel, it's protein in the plasma membrane that's opening and closing happily, it's sensing sodium, but it doesn't care what its neighboring channels are doing. And if that were the case, then each channel would open when it encounters sodium, and it would just open and close on its own. However, there's a mode of gating in which if a channel opens and it's nearby another channel, it may persuade the other channel to open. So it's a little bit like people in a room, but if they're by themselves, they're talking to themselves, they're not saying very much, but if they all get together, they all start shouting. And so this is cooperative positive gating, where one opens and they all open. So what I'm going to show you is that this happens in slack channels, and that it's much more prevalent in the mutations, and that can explain a large part of the gain of function. So now this is a wild-type channel, and now we're looking at openings going downwards, and these deflections downwards from the closed, the open 1, open 2 state. You can see this is a wild-type channel with 2 patches in it, and if you know how 1 or 2 channels behave, you can take a guess at what would happen if there were 4 channels in your patch. And you can, or 6 channels in your patch. You just simply add these sorts of things together and make a binomial prediction that if you have this sort of distribution for the number of closed, number open, and the number of doubly open states, this is what you would get if you had 6 open channels. But what Jack Cronengold in our lab found pretty quickly, because he was very disappointed when he looked at the mutations and couldn't find any gain of function, was that when you look at the activity of multiple channels, you find that this prediction that you get from having 1 or 2 channels doesn't hold. They're much more active when they come together in a cluster. So now, for a wild-type channel, you often see 6 open at exactly the same time, even though that would not be predicted from what you would see if you just saw a single channel. The most extreme example of this is in this particular mutation, which is actually near the core of the region. This was disappointing to Jack when he first looked at it, because he said, there's no way this is a gain of function. This channel has a smaller opening, lower conductance than the wild-type channel, and it's opening less frequently than the wild-type channel. And so you look at 1 or 2 channels, and this is the distribution you get with 1 or 2 channels. You would predict, if you happen to have 4 of them together, and they were just acting independently, this is the distribution you get. You'd never see 3 or 4 open together. You'd occasionally see 2 of them open together. When he then finds a channel, a patch with 4 channels in it, it's the exact opposite. They act like sheep. Most of the time, all 4 of them want to be open at the same time. Very rarely are they closed, and very rarely do you just see 1 open channel. So this is true for all of the other mutations that we had looked at. Okay, I'm going to take another pause now, because I'm going to move on to the animal models. So if anyone has any questions about that's probably the heaviest duty biophysics part of the talk. So I'm happy to explain anything about that part of the talk. And if there are no questions, it gives me a chance to take a gulp of water and carry on.
I'll ask a quick question at this point.
Okay. Yep.
Have any of the mutations changed the number of channels you're seeing inserted into the membrane, which would then change how many are maybe clustered together and able to participate in this cooperative gating?
None of the mutations have done that. So when Grace Kim actually did a bioaccumulation assay, where you actually label all the proteins in the plasma membrane, none of the mutations seem to alter the number that were in the plasma membrane, but it altered how they interact individually. Now, one of the interesting things is that we get these enormous increases in gain of function when we express them in oocytes or in cell lines. You will see that in real neurons, we get gain of function, but you don't get a 20-fold increase. And we think that is simply because when you express them in an oocyte or a cell line, you're actually forcing them together. So you're seeing that phenomenon of cooperative gating much more than you would in a neuron where you've got all the other channels and something is controlling exactly how many channels you have. Okay, so let me move to an animal model. So we made the mutation of R455H, the mouse mutant, which corresponds to the human R474 mutant. And again, when Imran Qureshi first looked at these mice, he was disappointed because he couldn't see any seizures, he couldn't see any phenotype. The heterozygous died, they were never born, but the heterozygous had no phenotype looking at them. But when he would put electrodes on the brain, actually, he then started to see that they were having seizures, but they were generally non-convulsive, so you didn't see them. So the mouse could still be running, it would stop and then continue to run, but there was a seizure going on there. And they would have these interatrial spikes. They also have problems with procedural learning. They don't learn the rotor rod as well as the wild-type mice. And as I think Al George asked earlier on, I think we're still looking at exactly what all of the behavioral phenotype of these mice is. But clearly, they're way better off than the humans because they grow, they reproduce. But that's, I guess, what being a mouse is. Okay, so I now need to make a point about potassium channels that even many neuroscientists don't fully appreciate. A potassium channel can either increase or decrease the activity of a neuron, and it depends on when it opens. So if you look at a neuron firing like this, if a potassium channel opens only during the peak of an action potential, its job will be to bring the membrane potential back, repolarize it much faster. And what that does is it prevents the sodium channels from inactivating, makes the action potential briefer, and allows the neuron to fire at much higher rates than it would if it hadn't returned the membrane potential that quickly. So all neurons, like interneurons and other neurons in the brain that are capable of firing at really high rates, they have selectively potassium channels in them that only open at these very positive potentials that are only encountered during the peak of an action potential. In contrast, if you have a potassium channel that opens near the resting potential, that has the opposite effect. If that channel opens, it will try to bring the membrane potential to a negative value even before you get to trigger an action potential. And that will make it harder to stimulate an action potential, and it will reduce the rate of neuron firing. And the reason for explaining that to you is because both of those things happen with the SLAC gain-of-function mutation, but they happen in different types of neurons. So Jean Wu in our lab recorded from excitatory glutamatergic neurons, pyramidal neurons in the cortex, and from GABAergic interneurons, from two different types of GABAergic interneurons. And he did this in culture, and he was able to record not only from the heterozygous animals, but because we can get embryos before they're born, he was able to get neurons from the homozygous mutation. And these are recordings of the electrical activity, and you can see that the potassium currents, which are the main thing we're seeing here, are bigger in the heterozygous than the wild type, and they're bigger in both the GABAergic and the excitatory neurons. In tiny little squares down here, if you plot the voltage at which these open, you'll see that in the excitatory neurons, they only start to open at very positive potentials near around about zero. In contrast, in the inhibitory neurons, they're already open at -40, -35 millivolts, much more negative potentials. And based on what I've just told you, the effect is predicted by what I told you. So these, where it only opens at the peak of an action potential, that allows the glutamatergic neurons, the excitatory neurons, to fire at much higher rates And so in the red, as you activate, depolarize the neurons, they fire at faster and faster rates compared to the wild-type neurons. In contrast, the inhibitory neurons fire at slower and slower rates compared to the wild-type neurons. And as you might imagine, that is very bad for maintaining the normal balance of excitation and inhibition. And we think this is the main reason there's intellectual spikes and seizure occurring in these animals. The excitatory neurons are too excitable and the inhibitory neurons can't fire fast enough to inhibit that. And so, full stop. That's what we think is going on electrically. But there's something else really interesting going on here. Look at the initial spike at the beginning here. This was sort of unexpected, but you look at this little spike when you first depolarize. This is the sodium current. And what you see is that in the excitatory neurons and the inhibitory neurons, which have different directions in their excitability, the sodium current is increased. And we can quantify that simply by measuring the sodium current, and it's increased in the HETs and the homozygous mutations. And then we can simply do biochemistry and look at the amount of sodium channels that are there. And you can see that simply by having introduced this mutation, in the heterozygous mice here, you have way more of this NAD1.6 sodium channel. We also have a little bit more NAD1.2. In the embryonic neurons and in the culture dish, you increase all of the three major types of sodium channels. They're increased. NAD 1.1, 1.2, and NAD 1.6. So just having this overactive potassium channel has caused the neuron to make more sodium channels. And since I've told you that it interacts with FMRP and it interacts with CYFIP1, that raises the possibility that it's the overactivity, the channel, that is increasing the synthesis of those channels. And so Taylor Malone, graduate student in my lab, went about testing the effect of SLAT channels on translation of messenger RNAs. And what he found was that simply take, again, a really boring, non-excitable cell like a neuron, an ATK cell or a CHO cell, and transfect it with a reporter for mRNA translation. So this is messenger RNA that would normally make actin, but we've replaced the actin coding region with a fluorescent protein. And we can see then the synthesis of new proteins simply by bleaching the cell and looking at how quickly it makes the new fluorescent protein. So let's look at the green line first. If you have SLAT channels, in there, but you don't stimulate them, and we have pharmacological ways of stimulating them with niclosamide, you get no increase in translation over not having the channel there at all. So if the black lines correspond to the rate of this synthesis, if you have no channel. If you stimulate the channel, however, you get a massive increase in the rate at which you start making this. And we've looked at this in a number of different ways, not just by fluorescence. And that's quantified here. The mutant doesn't need to be stimulated. It already is stimulating the translation almost as much as the fully activated wild-type channel. In these experiments, we didn't get any further stimulation when we stimulated the mutant, but in fact, in more recent experiments, we can actually stimulate even more the translation of proteins when we stimulate the mutant channel. So translation is out of control with this mutation. That was in a boring non-neuronal cell, but we've also looked at this in neurons, and this is a picture of translation, active translation going on in a wild-type neuron and in one of these mutant cells. We've used a technique called a proximity ligation assay, which I'm going to explain in a minute. But this shows proximity ligation with a compound called puromycin and beta-actin. What basically this is showing us is where new actin is being synthesized And you can see that it's being synthesized all over the neuron in the cell body and the dendrites. But if you count how much is being synthesized, there's a massive increase in the rate of synthesis in the mutant animal compared to the wild type. The bottom part of the slide just shows that in neurons as well as in these cell lines, if we stimulate SLAC channels, we get an increase in translation of the actin reporter, and it doesn't happen in the SLAC knockouts. Okay. Let me take one more quick break to see if there's any questions and then go into the home state. Okay. So how did this happen? Well, CYFIP and FMRP, the two SLAC interacting proteins, have a bunch of functions independently, but there's one thing they do together, and that is when they form a complex, they bind to this protein EIF4E, the eukaryotic initiation factor 4E. And when it's bound to this EIF4E, along with a bunch of other proteins and the messenger RNA that's associated with this complex, you can't initiate translation. In order to initiate translation, you have to remove these proteins, and then this EIF4E can bind to EIF4G, make the whole initiation complex, which binds to the small subunit of the ribosome, and then that can start the initiation of translation. So let's look at what that does, what mutation first does to the binding of CYFIP and FMRP to the initiation complex and to SLAC channel. What we really need to look at here is this little red box here on the left and on the right. On the right lane is the mutant. You see there's way more CYFIP1, way more FMRP, and much more phosphorylated FMRP bound to the mutant channel than there is to the wild type. Now let's look at the right. This is now looking at the interaction between EIF4E and these proteins. So the amount of CYFIP1 and FMRP that is bound to the initiation complex to EIF4E is greatly reduced with the mutant. It's a really striking effect. So things have translocated from the initiation complex to the channel when you have the channel mutation. This doesn't happen just with the mutation. It happens, but reversibly with the wild type channel. So now we're simply looking in the red box here at the wild type channel that's been stimulated and not stimulated. And you can see when you stimulate the channel, you get a marked increase in FMRP binding, and you lose the CYFIP and the FMRP that were bound to the initiation complex. They've now moved to the channel eddy. You've lost the interactions with that complex. Okay, so I'm just going to quickly explain the proximity ligation assay experiment. This is an experiment where you take two proteins. They don't have to be proteins, but they can also be other drugs. But you have antibodies against the two of them, and those antibodies are bound to DNA, and if these two DNAs interact, that can be amplified, and so you will only get a fluorescent signal if two proteins come together, or if, for example, if this is something, an amino acid or a pseudo amino acid that can be incorporated into a protein, if that's incorporated into a protein chain, you can measure new translation with this technique. So this is the technique we've been using to look at exactly the same thing. And what you see now is the interaction between FMRP and the initiation complex in a neuron with that's been unstimulated, and then after it's been stimulated, and you can see that there's a massive loss of the interaction of FMRP with EIF4E when you simply stimulate the wild type SLAC channel. This is the same thing you see in the inhibitory neurons. You get a loss of that interaction when you stimulate SLAC channels. But if you look at the PLA between SLAC and FMRP, or between SLAC and CYFIP, now you get a marked increase when you stimulate the channel. So this is showing you the same thing, that when you stimulate the channel, you lose the interaction between FMRP and the initiation complex. You've already lost it with the mutant, but you get a further loss when you activate that channel. And you have much more interaction of Slack with the mutant channel, but you can get a further interaction by stimulating these channels. So this is the model of what's going on. In the resting state, you have clusters of channels that when they become active, they recruit CYFIP1 and FMRP to the channel complex, and that liberates EIF3, and that can trigger the synthesis of actin. And I'm going to show you in a couple of seconds that it also triggers the synthesis of sodium channel subunits. There are probably many more. We're still looking at exactly what proteins are regulated by Slack, but at least these two we have definitive evidence for. So just at the very end now, I'm going to talk about the work we've done with antisense oligonucleotides. So as most of you probably know, these are synthetic oligonucleotides that when you introduce them into a cell, they can bind to a messenger RNA, and that will cause the degradation of that messenger RNA. So this is Slack messenger RNA. You will reduce levels of Slack just by destroying Slack messenger RNA, and reducing the levels of Slack should therefore release FMRP and CYFIP back to where they belong in the cell. So that we tested that. And indeed, that is what happens. If you treat neurons with an ASO against Slack, the interaction between Slack and FMRP goes way down, and that's exactly what you'd expect because there's no Slack to interact with the FMRP anymore. But strikingly, the interaction between EIF and FMRP becomes markedly stimulated. And this is quantified here for the mutant. So this is the mutant treated with the ASO. The interaction of the mutant with FMRP goes way down because you've lost the mutant protein, but the interaction with EIF4 and FMRP goes way up. We've also looked at the synthesis of the sodium channels using this proximity ligation assay, using a proximity ligation assay with puromycin, which is incorporated into nascent polypeptide chains, and these three different sodium channels. And what we find is that when you have the mutant, the R458 mutant, it's stimulating the synthesis of these sodium channels at a much higher rate than the wild type channel in each case. But when you treat with the ASO, you can reduce that quite substantially back to wild type levels. So the sodium channels are being reversed. I should say, I don't have time to show you, but we've also done the electrophysiology. The ASOs revert the firing patterns, the amplitude of the currents, and all of those things back to almost back to normal. There's some interesting things with the inhibitory neurons. So the penultimate slide. So in work with Tim Yu, we've actually were able to treat 2 patients with these ASOs, and the ASOs were able to reduce the intensity of the seizures in both patients quite substantially. That is still ongoing, I understand, for one of the patients. But we're also able to show that in the human-derived iPS neurons, they also have a gain of function, and that could be reversed with the antisense oligonucleotides. They also reverse the sodium currents in those nucleotides. So this is still an ongoing, obviously, something we're all very interested in about these gene therapies. So I'm basically at the end of my talk, and the normal thing to do now will be to say, yep, so this is the summary that we know that this is now, this channel is a regulator of protein translation. It's regulating the ability of FMRP and CYFIP1 to suppress translation. And that's it, and show a summary slide. But I'm not going to do that. I'm just going to show one more slide on something that we have no understanding of at all, but it's really intriguing. So we have done EMs on the slight knockout, the wild type, and the gain of function. And we're prompted to do this by a proteomic study that we did in collaboration with Biogen. And we found that in the knockout, as well as in the knock-in, there were major changes in proteins of the inner mitochondrial membrane. And so what we found was that if you simply look at the mitochondria in the wild type, there's a wide variety of cristae configurations. But the density of cristae, it takes over a wide range of cristae. But if you look at the knockout or the knock-in, they have much denser cristae. These are the original images. This is where we just outline the cristae in digital form on the right. But this is highly statistically significant. And not only is it statistically significant, but the technician who was doing this could identify whether it was a knockout or a knock-in compared to the wild type simply by looking at the pictures of the EMs of the cortical neurons. So somehow SLAC channels, KCNT1 channels, are hooking up to metabolism. We speculate this is a part of what's underlying the problems in the synaptic plasticity, but that this is a wide open area. So I'll leave you to think about that because I'm still thinking about it, but it's a puzzle. And finally, these are the people who've worked with. Jean Wu is very important in much of the work on the proximity ligation assays. Taylor Malone showed that the channel actually regulates translation, and all of these other people are doing major contributions to the ongoing work. We collaborated with Tim Yu as well as others on the human cells. And I'm going to stop there because I think I'm just about there in time.